Assalamu alaikum. Hello everyone, I'm Awab Musaad, and in this hopefully short video, I'll be talking about number of translocations. And before I do so, just want to say Ramadan Kareem to everyone, and let's begin. So the translocations mentioned in Dr. Firdaus' lecture are divided based on the disease they are related to. So some translocations that causes leukemia, and others that causes non-Hodgkin lymphoma, and I'll be talking about them based on this. So just to establish the basis that we know that translocation is basically a process where the chromosomes exchange their genetic material, and sometimes we'd have a combination that is basically unwanted that facilitate the development of cancers. Right? So let's start with the translocations that causes leukemias. Okay, the most famous one, which is the Philadelphia chromosome, caused by translocation between the ABL gene, which is a proto-oncogene that encodes for the tyrosine kinase, a very important enzyme for the proliferation of cells, on chromosome 9, and the BCR gene on chromosome 22. And based on Dr. Firdaus' lecture, the function of that is unknown. Okay, so they will lead to the fusion, as we said, forming the Philadelphia chromosome. And you can guess, since this is a proto-oncogene, that means the fusion lead to the overexpression of this. So more ABL expression, more tyrosine kinase, and more proliferation of cells, leading to chronic myelogenic leukemia most of the times. Okay, and the second translocation I want to talk about is the Ato gene in chromosome 8, and this gene, by the way, encode for a protein called the A kinase anchoring protein, and it's translocated with another gene called the AML1 gene, which is normally a regulator. That means that it make sure that the cells differentiate, proliferate normally. So that's we can say it's a tumor suppressor. Okay, so the fusion or translocation between those two will lead to the inhibition of this one. Okay, meaning now we'd have a less regulated cells, right? And the last translocation I'll talk about regarding the leukemias is between the Pml gene, okay, in chromosome 15, which is basically a protein, a gene that encodes for a protein called the promyelocytic leukemia protein, and another gene which is called the retinoic acid receptor alpha gene, which is normally a tumor suppressor on chromosome 17. And also you'd guess that the fusion between those, meaning that it will inhibit this tumor suppressor. Okay, so now the cancer cells would be able to proliferate more freely, right? Leading or facilitating the development of leukemias. Right? So let's switch gears to the non-Hodgkin lymphomas. So there's this gene called the IGH gene. Okay, basically it's a gene that in chromosome 14, it encodes for the heavy chains in the antibodies. Okay, the IGH stands for the immunoglobulin heavy locus, and basically it's quite troublesome because translocation between this gene and 4 different genes in 4 different chromosomes lead to 4 different types, or let's say lymphomas, classified as non-Hodgkin lymphomas. Okay, so translocation between the IGH gene and the BCL6 gene, okay, on chromosome 3. Normally the BCL6 gene is inhibit the differentiation. Okay, so it's a proto-oncogene, right? The cancers they proliferate and they don't differentiate, right? So the translocation or fusion between those two lead to the overexpression of this gene, okay, leading to diffuse large B-cell lymphoma. Okay, and the translocation between the IGH gene and another gene called the Cmyc gene, it's a proto-oncogene, and it's in chromosome 8. Notice how it's in chromosome 8, but it's a different gene than the ATO, also in chromosome 8. And so since this is a proto-oncogene, the fusion between those two will most likely, or basically, overexpress the Cmyc gene, leading to the Burkitt lymphoma. Okay, and when the IGH also translocate with the cyclin D gene, chromosome 11, which is proto-oncogene as well, it will be overexpressed, leading to mantle cell lymphoma. Okay, and last but not least, when the IGH gene translocate with another gene called the BCL2 gene, okay, which is in chromosome 18, normally it's a tumor suppressor. Okay, so we'd guess, you know, when those two genes translocate, this gene will be inhibited, right? So tumors will be able to proliferate freely, right? And that normally the translocation between those two genes lead to follicular lymphoma. Okay, and that's basically it. Feel free to leave feedback, and thanks for watching. Assalamu alaikum.